I found this topic quite daunting, and the title a little even more so. But fortunately for you and me, we won't be talking about all 700 syndromes. We'll be going really over more of a historical overview of the general path to discovery of causes of autosomal intellectual disability. So, over 800 genes have been linked to either isolated, non-syndromic forms of ID or to a disorder that includes ID as its major feature. And this is from a Nature Reviews article from early in 2016 showing data from the previous year. So this data is a little bit behind, but the curves give you an idea of the general trajectory of discovery. And as you can see in the green, this is the X-linked associated ID genes, which reached around 100 and have plateaued, as you would expect, with the limited number of genes on that chromosome. But the numbers of autosomal genes associated with ID has historically lagged, but has continued to make headway more recently. So the genetic diagnosis of ID first began with the discovery of trisomy 21 as the cause of Down syndrome. It was in 1959 that Jerome Lejeune reported his findings to the Academy of Sciences Journal, providing evidence to support that 3 copies of chromosome 21 cause Down syndrome, and so coining the term trisomy 21. And with the development of cytogenetic banding techniques, chromosome abnormalities were soon recognized as a common cause of ID, explaining up to 15% of cases. So clinically distinct ID syndromes became recognized, including Prader-Willi syndrome, Angelman, Williams, Smith-Magenis, Miller-Dieker, and velocardiofacial syndrome. And as we know from Dr. Schwartz's talk, chromosome X became the main focus of research in the 1990s and has continued. There have been significant barriers in the research for autosomal ID. The challenges included the tremendous size of our genome, the limitations of cytogenetics and sequencing technologies, and the absence of large families with autosomal forms of ID, particularly those with more severe ID. Those individuals are having children. So the use of genomic microarrays, with its increasing availability over the last 10 years, has allowed for genome-wide detection of copy number variants at a higher resolution than possible under the microscope. And from larger microarray studies in individuals with ID, we learned that CNVs occur de novo in the germline of about 10% of these patients. And with the superior resolution over conventional karyotyping, genomic microarrays have become first-tier testing for ID. There are numerous publications describing recurrent CNVs and discussions investigating their significance. And this article, Copy Number Variation, Morbidity Map and Developmental Delay, is from Nature Genetics in 2011. And they compared CNVs in over 15,000 children with ID and with various birth defects to 8,000 adult controls. And from their data, they estimated up to 25% of children, or affected children, carried CNVs over 400 kb, compared with only 11.5% of controls. And they reported 59 CNVs accounting for 10% of patients, and then more candidate genes were identified. And so as a clinician, some of these array results have become great pieces to help solve the puzzle in understanding intellectual disability, but also raise a lot of questions with the variants of uncertain significance that we find, or the normal results where we still don't understand the cause of ID. So moving forward, with the development of next-generation sequencing and exome sequencing cohorts of individuals were being tested, those with similar phenotypes. And this led to numerous articles being published for the identification of disease-causing genes. And a few included those in 2010 when this really picked up for discovery of genes associated with Schinzel-Gideon, SETB-P1, and one of the genes for Kabuki syndrome, MLL2, was identified, as well as the ASXL1 gene associated with Oren-Opitz syndrome. For non-syndromic ID, or cases where features were non-specific, multiple approaches have been taken. And in the Western world, de novo or heterozygous mutations and copy number changes that are felt to be the majority of the genetic forms of ID. So new changes. So trio-based approaches have begun with parents and proband, and trying to find that de novo dominant mutation causing sporadic forms of ID. A trio-based approach is often what we might take for our patients with unknown diagnoses in clinic, and we're finding answers for cases where features are subtle or not so classic, but in hindsight fit the clinical features associated with the whole exome finding. And this is an article from Human Mutation in 2015. Grozeva and the other authors were looking at strategies to identify genetic causes of ID. And they screened 986 individuals with moderate to severe ID for variants in 565 known or candidate ID-associated genes using targeted next-generation sequencing. In total, that panel consisted of 253 known and 312 candidate ID genes. And control samples from 905 individuals with congenital heart defects were used. They found that likely pathogenic rare variants were detected in 11%, or 113 variants in 107 of the 986 individuals. This was 8% of individuals who had loss of function variants and 3% who had known pathogenic missense variants. Variants in SETD5, ATRX, Col4B, MECP2, and ARID1B were the most common causes of ID, where ATRX, Col4B, and MECP2 are all X-linked. And this is just a slide for SETD5. This is a gene on the chromosome 3, and individuals typically have moderate to severe ID with variable features. So even the photos, the facial phenotype seems to be variable. But they have brachycephaly, prominent high forehead with synophorus, a long tubular nose, narrow palpebral fissures, and fleshy low-set ears. They can have obsessive compulsive features and ritualistic behaviors and autism. And we've heard a lot about ARID1B in our center. It's on chromosome 6. And individuals have developmental delay and intellectual disability with growth delay. And phenotypes include those individuals who have classic phenotypes for Coffin-Siris. They have also been linked to Nicolaides-Baraitser and non-syndromic ID as well. So understanding that not all children have biological parents available to complete a trio exome analysis, but still finding utility in the molecular testing, the authors from this 2015 article recommended that a strategy of investigating all loss of function variants and known genes that cause ID, as well as assessment of a limited list of proven pathogenic missense variants and genes would be helpful. And they observed that this strategy provided an additional 11% yield above the 10 to 15% yield by array alone. So in other populations, the investigation of autosomal recessive genes associated with ID has made more sense due to their relatively higher rates of consanguinity. And from 2011, this is an article that was published. It was one of the first systemic studies of genetics underlying recessive intellectual disability. using homozygosity mapping and targeted NGS to identify many new candidate genes. And they looked at homozygous regions in 136 consanguineous Iranian families with autosomal recessive intellectual disability. And they identified recessively inherited mutations in 23 known genes, and 50 new candidate genes were identified. They found probable causative mutations in 78 of the 136 families, and that's 57%. In 2015, another group reported on exome sequencing studies of 143 large consanguineous Saudi Arabian families, and their studies revealed 33 novel candidate genes involved in different neurological disorders. We know that microcephaly is associated with higher risk for intellectual disability, and primary microcephaly is rare in some populations, maybe 1 in 30,000 to 1 in 250,000. But the incidence in regions where marriage between cousins is common, it can be as high as 1 in 10,000. And from here you can see where there have been genes identified in some of these populations. Investigations of consanguineous populations have led to identification of at least a dozen genes causing primary autosomal recessive microcephaly. And this is just a picture of a child who was found to have 2 mutations in the ASPM gene, and that's the abnormal spindle-like microcephaly-associated genes. And these investigations and further research have allowed for a successful creation of mammalian models, allowing for better understanding of the pathophysiology for primary autosomal recessive microcephaly. And recent evidence suggests that the primary disorder is in neurogenic mitosis that causes the decrease in brain size. So with these advancements in technologies and increasing diagnostic yields, we're still faced with significant challenges. Intelligence is really a complex trait. And when we find ID as part of a de novo syndromic pattern of unique physical characteristics, or ID in a family where clear autosomal recessive inheritance pattern is seen and physical features are specific, we think intuitively that typically we might find a change identifying a cause more likely. But in this bottom pedigree, in a non-syndromic ID, or where there are multiple family members on both sides of the family with mild learning problems or borderline learning problems or milder intellectual disability, it's really hard to find a cause that's clear. The number of autosomal dominant ID genes have remained relatively small, but is growing, and over 300 autosomal recessive genes associated with ID have been found. And this is mostly through homozygosity mapping via SNP arrays and follow-up sequencing of candidate genes. Most are probably syndromic, with a minority of genes causing isolated autosomal recessive ID, and that number of candidate genes will continue to rise as whole exome and whole genome sequencing is completed. Having candidate genes still leads to additional challenges of proving pathogenicity, and we know that there are specific guidelines that have been published on how findings are reported. Either they're VUSs, likely pathogenic, or pathogenic variants. But even so, with these categories, it can be difficult in the clinical setting to correlate these symptoms and interpretation. We ask ourselves, are there other factors, genetic factors, environmental factors? And there's also a challenge of replication of results and finding enough phenotypically similar patients to test. And this is particularly difficult for those with non-syndromic ID. So additional databases and repositories are becoming essential in collaborations. And these are just a few, including those from Phenome Central and Matchmaker, Exchange, and GeneMatcher. And I know that the lab has really utilize some of these. Groups are publishing on ways to provide systematic, curated, ID-associated gene catalogs with core phenotypes and clinical classification systems. And to me, this is still very complex, even though this is supposed to make things more clear. But I think over time, maybe we'll see all these connections and how they work and put things into better perspective. So we'll likely hear more about functional studies with stem cells, as well as the use of CRISPR-Cas9 gene technology. And together, these may allow for more accurate characterization of functionality. And I'll close with the next couple of slides. All in all, there really has been a significant rise in discovery of genetic causes of ID, especially over the last several years. And this is a graphical overview of the diagnostic yield for moderate to severe ID, excluding Down syndrome. And the solid line shows the mean of the published studies' diagnostic yield, and the shaded areas shows the upper and lower boundaries. And as you can see, in the 1970s, it's pretty low, and this is where conventional karyotyping became a routine diagnostic test and provided diagnosis in about 3 to 7% of cases of ID. Then, with technologies of Sanger sequencing and FISH, we see another small jump in the late 80s to 90s. And with genomic microarrays in the early 2000s, diagnostic yield increased by another 15 to 23%. And whole exome sequencing in 2010 added a diagnostic yield of 24 to 33%. And some of the first pilot studies using whole genome sequencing in 2014 added another 26%, which led to a diagnostic yield of 55 to 70%. So there is a significant discovery in autosomal ID, but we may just be on the cusp. We still have a big, long way to go, but I think we're all here and optimistic and excited for all the new discoveries. So I'll close with that. I'll take any comments or questions.